Guys, welcome to my YouTube channel. This is me, Hasnat again, and today I'm going to perform experiment. We are going to extract DNA from mice stool. So I'm going to show you step-by-step procedure, and we are going to follow this protocol. I'm going to mention this protocol link and name in the description. If some research student want to perform the same experiment, this video will gonna help you in this. So say me thank you later. And I have my friend here, she's also going to help me because I have so many samples, so it's really hard for me to perform all of them by myself. So we are going to do it together. Yes, let's. So here are my samples, as you can see. So the next step is we are going to weight of these samples. It must be 0.25, less than actually. Okay, so it's 0.21, so the weight is fine. So I'm going to use this fast DNA stool mini kit. We have to add 1 ml InhibitEX Buffer to each sample. So let's do it. Before we start our experiment, we have to adjust this temperature to 70. So I already adjust and now it's heating up, so it will go high to 70. We are going to boil our sample in this so it get mixed properly. After that, we are going to add InhibitEX Buffer 1 ml to our sample, each of them. I'm going to add one by one. Almost done. It's done. All samples. No, still. So I'm going to say one more thing. Like this sample, it's not fully dissolved. Even I did Botox for more than 5 minutes, but it has to be dissolved. But because it's not dissolved, so I'm going to use the tip of pipette and dissolve it fully, so we can recover the maximum DNA, maximum concentration of DNA. So this is just a personal idea. It's not mentioned. It's not mentioned in the.
Smell is terrible.
Yeah, the smell is terrible. Of course, it's stool. I dissolve my all sample with the help of pipette and with the help of Botox. I'm using this as a stand to put into hot water. So my colleague almost done. Finish? Okay. So now the temperature, and I already put in here. So let it rest for 5 minutes.
Doing that is okay.
So now our samples are still heating, and we are going to use this column, the spin column. So I'm going to prepare this in advance, and I'm going to add 1.5 microliter proteinase A. So let's do it. It's okay. Down. We transfer 200 supernatant from these tubes to this tube. Now we are going to add almost exactly 200 buffer AL, and then we do Vortex. So I already added 200 microliter AL buffer, and now you can see that it is separate. I mean, it's on the upper layer from here. Now we will mix it before doing incubation for 10 minutes at 70 Celsius temperature. After incubation for 10 minutes, I'm going to add 99% ethanol, 200 to the Vortex. So after we transfer our solution from here to columns, now we are going to centrifuge the column for 1 minute. We are just doing DNA extraction and look at our table. Now I'm going to just take the column and then transfer it into new column tube like this. So even after centrifuge, I did the column centrifuge again when I put it in new tubes, and we can see there is a little bit liquid inside, it's ethanol, and ethanol can disrupt our results. So this step is up to you. If you want, you can do it twice or just one time. Like after you did the previous step, the next step is to just centrifuge empty tubes so we can get more solution. So now again I'm going to transfer it into new column, and then we are going to start our washing process, and we are going to use AW1 buffer 500 microliter in each tubes, and then centrifuge again. So now we did the centrifuge, and I'm going to transfer just the column to new tube, and now we are going to wash all of this with, and then centrifuge again, the same step but with new solution A2. The centrifuge, I'm going to transfer the column to here in EP tubes like this, and just take the column out and transfer here. And now I'm going to add 200 buffer ADE to these tubes, and then we will let it stay for 1 minute, and then we centrifuge. Now let it rest for 1 minute. 세게 세게. 아니야. 세게 세게 여기. 이렇게. 이런 거. 지금 생. 지금 얼마까지 있어요? 오케이. We take the column out, and we have our DNA in this solution, so we are going to check it using the nano drop machine. Wow, it gives music. It's like a song. First a manual, and we are going to check the concentration of our DNA, but first we will take a blank, which is just a DW. And arrange your sample, like my sample have week 1 and week 2 and some control samples, so I'm going to arrange that. So arrange the samples, and now we are going to check the concentration. So my DNA sample concentration is really low, like 21 and 14, but that's fine because it's a wild samples. So I'm doing this experiment for the purpose in which low concentration is also fine. So like this you can do and check the concentration all of your samples. I hope you like this experiment. I mean, this experiment will be helpful for you. So any students out there doing research, if you like this video, gives me thumbs up, and please share and subscribe. See you in the next video. And she did really well. Cheong, you did really well. So bye bye. See you next video. Bye.